Good morning, everyone. Hello and welcome to this online lecture on muscle and nervous system histology. In this lecture, we will explore the microscopic structure and function of two of the most important systems in the human body, the muscular system and the nervous system. By the end of this lecture, you will have a deep understanding of the intricate and interdependent workings of these two critical systems and their importance to our overall health and well-being. Through this presentation, we will explore the fundamental components of muscle tissue and the organization of muscle fibers, as well as the structure and function of the nervous system and the specialized cells and structures that allow us to perceive, interpret, and respond to the world around us. Um, with that, let's get started and dive into the world of muscle and nervous system physiology. Muscle histology can be divided into two main categories: striated muscle and smooth muscle. Striated muscles are characterized by a striped appearance under the microscope due to the presence of alternating light and dark bands, and we call these bands striations. These muscles can be voluntary, as is with the case of skeletal muscles, or they can be involuntary, as is with the case of cardiac muscles. Skeletal muscles are attached to bones and it's responsible, as we know, for the movement of the body. Cardiac muscle is found only in the heart and is responsible for pumping blood throughout the body. Both types of striated muscle, whether skeletal or cardiac, are composed of long cylindrical cells called muscle fibers. They also go by the name of myofibers. These myofibers have multiple nuclei and are organized into bundles called fascicles. Within the myofibers, these striations are because of the arrangement of certain contractile proteins known as actin and myosin. Okay, and what facilitates muscle contraction is the sliding of these actin and myosin myofibers past each other. Now, smooth muscle, on the other hand, is characterized by the absence of striations, unlike striated muscles, and is found in the walls of hollow organs such as the intestines, blood vessels, the uterus. It is responsible for controlling the flow of fluids and food and other substances through these organs. Smooth muscle is composed of spindle-shaped cells, and unlike striated muscle, each cell contains only a single nucleus. Unlike striated muscles, the contractile proteins in smooth muscle are much more disorganized, and as mentioned, there are no striations. Contraction in smooth muscle is regulated by the autonomous nervous system and by hormones and are thus involuntary. Okay? In summary, muscle histology can be divided into striated and smooth muscle, which is differentiated by the presence of um, these striations in the cells. Now, this slide shows us the histology of the three different types of muscle tissue. Here you have smooth muscle, okay? You have skeletal muscle fibers here, and then this is cardiac muscle. Okay, please take note that cardiac and skeletal muscles have these striations that we see here, which smooth muscle does not. Skeletal muscle is enveloped by connective tissue for several reasons. Number one, it serves as protection. Okay? So the connective tissue surrounding skeletal muscle provides protection to the muscle fibers by protecting or cushioning them from external forces and preventing damage from physical trauma. They also play a part in structural support. The connective tissue investments, also known as fascia, provide structural support to the muscle fibers and help maintain their positioning and alignment relative to each other and relative to the bones. This allows the muscle to generate the force more efficiently and effectively so that the body can move. These connective tissue wrappers also serve to play a role in force transmission. Connective tissue serves as a conduit for the transmission of forces from the muscle to the bone where it is attached. This allows for the transfer of force across the joint, and this, of course, facilitates movement. The connective tissue surrounding the skeletal muscle contains blood vessels and nerves that supply the muscle fibers with oxygen and nutrients and transmit signals for muscle contraction and relaxation. So they also play a very important role in vascularization and innervation. Okay? And these connective tissue investments are known as epimysium, which surrounds the entire muscle, perimysium, which surrounds groups of muscle bundles or fascicles, okay? And then you have the endomysium, which surrounds individual muscle fibers. Okay? So again, there are three main layers. You have the epimysium, okay, which is the outermost layer that surrounds the entire muscle. Okay? So this is the epimysium. It is composed of dense, irregular connective tissue, as what we have talked about last time, and provides structural support and protection to the muscles. Okay? A bundle of muscle fibers, known as a muscle fascicle, is surrounded by the perimysium. So this is the middle layer. Okay, so and epimysium is the most external. The second layer is the perimysium, and again, it is composed of dense irregular connective tissue. And what it does is it actually opposes or wraps nerves and vascular connective tissue or blood vessels to the muscle fascicle. So it plays a very important part in innervation and vascularization. Now, the endomysium is the innermost layer, and it surrounds each individual muscle fiber. So each individual muscle cell is surrounded by an endomysium. Okay? In contrast to the epimysium and the perimysium, which are composed of dense, irregular connective tissue, the endomysium is composed of loose connective tissue. Okay? And in the same way that the perimysium opposes larger blood vessels and nerves to the muscle fascicle, the endomysium opposes smaller nerves and capillaries to the individual muscle fibers. Now, skeletal muscle cells, and we know that they are also known as muscle fibers or myofibers, have a very distinct histologic appearance. They are elongated, cylindrical cells, and they can range in length from a few millimeters to several centimeters. It depends on the muscle where they belong to. Now, here are a few key characteristics that will differentiate skeletal muscles from the other different types of muscle cells. Number one, skeletal muscles are multinucleated. Okay, this means that they have multiple nuclei along their legs. Okay, these nuclei in the skeletal muscle are not located centrally. Instead, they are located just beneath the cell membrane. Okay, the cell membrane of the muscle is known as a sarcolemma, and it is responsible, as with the nuclei of other cell types, they are responsible for controlling cellular metabolism and protein synthesis by the production of DNA. Now, another key feature, as we've talked about earlier about skeletal muscle cells, is that they are striated, okay? Which means that they have alternating dark and light bands when they are viewed under a microscope. This banding pattern or this striation pattern is due to the arrangement of contractile proteins known as actin actin and myosin. Okay? And these actin and myosin filaments are organized into units called sarcomeres. Okay? And that's what we will be talking about later. Sarcomeres are the basic functional units of the muscle fibers. Okay? Now, as mentioned, um, the cell membrane of skeletal muscle is known as the sarcolemma. Okay? It is a specialized cell membrane that is capable of generating and transmitting electrical impulses, which are known as action potentials. I think you've already talked about this in your physiology classes. And these action potentials are necessary for the initiation of muscle contraction. Okay? Now, in the same way the cell membrane of skeletal muscle is specialized and again are known as sarcolemma, the cytoplasm of skeletal muscle is known as sarcoplasm. Okay, again, the cytoplasm of skeletal muscle cells are known as sarcoplasm. As usual, it contains numerous organelles, the mitochondria, the endoplasmic reticulum, same as with any other cell type. Now, skeletal muscle cells have specialized structures known as transverse tubules or T-tubules, and they are actually just invaginations of the cell membrane that extend deep into the muscle fiber. So they are invaginations of the cell membrane protruding into the muscle fiber. And they serve to allow for the rapid transmission of action potentials, okay, which would allow for synchronization of the muscle contraction. Because for the muscles to be able to generate adequate force, um, the muscle contractions must be synchronized with one another. Okay. Now, the myofibrils are specialized structures or organelles that are found within skeletal muscle cells. Okay. Please note that myofibers are the muscle cells, myofibrils are organelles, and these represent um, contractile proteins, specifically actin and myosin. Okay, the thick filaments are known as myosin, and the thin filaments are known as actin. Myofibrils are arranged parallel to each other along the entire length. Okay, and they are very highly organized. So this parallel arrangement is very important because it facilitates, again, the strength of the contraction. Okay, if the muscle fibers or if the myofibrils are not organized that way, then you will not generate enough force to facilitate contraction. Okay, so the organization of these myofibrils within the functional units, again, the functional units are known as sarcomeres, is highly regulated, and they are also very highly coordinated by a number of regulatory proteins, including tropomyosin and troponin. Okay? These proteins help to control the interaction between actin and myosin and ultimately regulate the rate and intensity of the muscle contraction. So it's not just the actin and myosin interacting with each other. You also have smaller proteins known as troponin and tropomyosin, and they regulate the interaction between actin and myosin. Okay? So each myofibril is composed of repeating units called sarcomere. And as mentioned, sarcomeres are the functional units of skeletal muscle fibers. Okay? Sarcomeres extend from one Z line to another Z line. Okay? So sarcomeres, again, are composed of thick filaments known as myosin and thin filaments known as actin. And um, they slide upon each other during muscle contraction, and this results in the shortening of the muscle fiber and the sarcomere, which results to the contraction of each individual muscle fiber. So, if you look at skeletal muscle under a microscope, you will actually see distinct bands. And again, these are seen only under the microscope. Okay? Now, first things first is we need to define what a sarcomere is. A sarcomere, again, is the functional unit, and it extends between adjacent Z lines. So this dense band here at the edges, this one is a Z line, this one is a Z line. Okay? So one Z line here, another Z line here. Okay? Now, the area between two adjacent Z lines is known as the sarcomere. This is a functional unit or a contractile unit of the muscle fiber. Okay? Now, in the middle here, you have what is known as the A band. A band or the anisotropic band. Anisotropic band. Okay? This is also known as the dark band because it stains darkly. The reason why it stains darkly is because it serves as the area where the thick filament or myosin and the thin filaments or actin overlap. So that's why it's dark because diri mag-overlap ang thick filaments or myosin and thin filaments. Again, this is the dark band or the A band. Okay. Now, the area between the Z line and the A band is known as the I band. I means isotropic band. Okay, and it stains slightly. It stains slightly because only the actin filament or the light filaments are located here. So that's why it tends to stain lightly under the microscope. Okay. Now, the H band or the H zone is the area where is the area within the Z band wherein the actin filaments no longer cross. Okay, the H zone is the area of the A band where the light filaments do not reach during the uncontracted state. Okay, so as you can see here, if the muscle is in the relaxed state, you will have an area in the middle where the actin filaments do not reach. Again, this is only seen in the relaxed or uncontracted state, and you call this the H zone. Okay? The H zone disappears when the muscle is contracting because the actin filaments will slide towards the middle. So the H zone will only be present when the muscle is relaxed. It disappears when the muscle is contracting. The area in the middle of the M line of the H zone is known as the M line. And when a muscle contracts, this represents where the actin filaments will contact each other when the muscle is in state of contraction. Okay, so again, Z bands, which represent the ends or the limits of the sarcomere. You have the myosin band or the heavy chain. You have the actin chain. Okay, the area that is where the actin and myosin filaments overlap is the A band. Okay, the area between the Z line and the A band is known as the I band, and this represents the actin filaments. Okay, the area in the middle of the A band, which is seen only when the muscle is uncontracted or in a relaxed state, is known as the H zone. In the middle of the H zone is the M line, and this is where the actin filaments will meet when the muscle is contracting. Okay. So sarcolemma, this is the parts of the skeletal muscle fiber. You have the sarcolemma or the cell membrane. You have invaginations, which are known as T-tubules. Okay. You have specialized endoplasmic reticulum known as sarcoplasmic reticulum. Okay. Here are photos of skeletal muscles. Okay, so here you have longitudinally arranged muscle fibers. Okay, you might notice that the bundles of cells here are quite different. Well, when compared to this, it's because um, if you're looking at it, you're actually looking at the side view. This one, you are looking at it the cross-center view. Okay, so this is the inner circular layer, the outer longitudinal layer. If I'm not mistaken, this should be located in the esophagus. Okay, now please take note that you have a lot of nuclei. They are multinucleated. Okay, and the nuclei are located in the periphery of the cells. Okay? The muscle is composed of myofilaments, actin, troponin, myosin, and fibromyosin. And they are all important proteins involved in muscle contraction. They are located in specific regions within the sarcomere. Okay, so actin is a thin filamentous protein that is located in the eye band. So the actin filaments are here. Okay, and they are anchored at the Z discs at the ends of the sarcomere. Okay, now troponin is a complex of three proteins. Okay, so it's composed of troponin P, troponin I, and troponin C, and they are attached to the actin filaments. Okay, troponin regulates muscle contraction by binding calcium, which causes a change in the structure on the troponin tropomyosin complex, allowing the myosin heads to come into contact with actin filaments. So, troponin, um, in conjunction with calcium and tropomyosin, serves as the key which will facilitate the interaction between actin and myosin. Okay? So, as mentioned, tropomyosin is another protein and it is attached to the actin filament, and it acts as a regulatory protein. It inhibits the interaction between actin and myosin. Okay, so for there to be interaction between actin and myosin to facilitate contraction, tropomyosin must be deactivated, and to deactivate tropomyosin, troponin must bind to calcium so that it can signal tropomyosin to be unlocked. Okay, and thus facilitating the interaction between actin and myosin. Okay. Now, as mentioned, myosin is a thick filamentous protein. It's a thick protein that is located in the A band or the anisotropic band in the sarcomere. Okay, the myosin filaments overlap with the actin filaments, and this is known as um, the the anisotropic band. Okay, okay. So at the molecular level, skeletal muscle is composed of repeating units called sarcomeres, and these are the basic functional units of muscle contraction. Okay, I keep repeating that because that is important. Now, each sarcomere is composed of thick myosin filaments and thin actin filaments that are organized again in a highly ordered manner. Okay, so the region in where the myosin filaments are arranged is known as the A band, okay? And the actin filaments are located to the sides, okay? The actin filaments, as mentioned, are anchored at the ends to the Z discs, okay? Muscle contraction occurs when the myosin filaments slide past the actin filaments, which pulls the Z discs closer to each other, and this facilitates the shortening of the sarcomere. Now, this sliding is powered by um, the hydrolysis of ATP, and this is regulated by the binding of calcium ions to troponin. Okay? So overall, the highly organized molecular structure of skeletal muscle allows for very precise control of muscle contraction and the generation of force of movement. Okay? So this is how it will look like. So you have the myosin chain here, and then you have the actin chain here, together with the regulatory protein. So note that the myosin chains have these heads, and they serve as rudders, and they serve as paddles to pull the actin filaments closer towards the center. And then as the actin filaments is pulled by myosin towards the center, the Z discs will come closer to each other, facilitating muscle contraction. This is how it will look like. Okay. Now, please note that ATP plays a role in muscle contraction because it facilitates the release of the myosin heads from the actin chain. Okay, so ATP plays a role in the release of the myosin heads from the actin chain so that it can pull it closer towards the middle. So again, I would like to point out that ATP plays a role in muscle relaxation, okay, or in the release of the myosin head from the actin chain, okay? So, skeletal muscle contraction is initiated and regulated by a complex series of events that involve the interaction of a lot of substances and ions. Okay, so it starts off when a nerve impulse or an action potential travels down a motor neuron and arrives at the junction between the neuron and the muscle. This is known as the neuromuscular junction. Okay? When the action potential or the nerve impulse reaches the neuromuscular junction, it facilitates the release of a substance known as acetylcholine into the space between the neuron and the muscle, and the space is known as the synaptic cleft. Okay? Now, acetylcholine will then bind to the receptors on the sarcolemma, and it will facilitate depolarization and generation of an action potential in the muscle fiber itself. Okay? Now, once the sarcolemma is depolarized, the action potential travels from the sarcolemma into the invaginations known as the T tubules, and they carry the action potential deep within the muscle fiber. Okay, this facilitates the movement of the action potential from the sarcolemma through the T tubules through the sarcoplasmic reticulum, which is a specialized endoplasmic reticulum. Now, the sarcoplasmic reticulum is specialized in that it serves as the reservoir for calcium in the muscle cell. Okay? Now, once the action potential reaches the sarcoplasmic reticulum, calcium is released from the sarcoplasmic reticulum into the cytoplasm. Okay? And this will facilitate um, contraction. Okay? Now, in the normal resting state, Okay, we know that actin is prevented from interacting with myosin due to the presence of tropomyosin. Okay, now when the calcium ions that were released from the sarcoplasmic reticulum starts to attach to troponin C, okay, this will facilitate a change in the structure or conformation of the troponin tropomyosin complex. Okay, and they will facilitate the exposure of the actin to myosin. Okay, so once calcium is released, it will bind to troponin. Troponin will change the conformation of the troponin tropomyosin complex. They will hide, and instead, actin will be exposed and will be free for interaction with myosin to facilitate contraction of the muscle. Now, we know that um, once the myosin heads are exposed to actin, they will form cross bridges. Okay, they will link to each other, and they will start to pull um, the muscle cells slowly towards the middle. Okay, now ATP comes into the picture because when ATP is hydrolyzed, um, once the myosin heads have pulled the actin filaments toward the center, hydrolysis of ATP will facilitate to reset or release the heads from actin. Okay, so you will have this ratcheting mechanism that slowly pushes the actin filaments towards the center. Okay, and this process wherein the actin filaments are pulled towards the center and the release of ATP and energy will facilitate the release of actin from myosin, and then another cycle will continue. This is known as the power stroke. Okay, when the action potential ceases, calcium ions are pumped back into the sarcoplasmic reticulum, and because calcium levels become low in the cytoplasm or the sarcoplasm, the troponin tropomyosin complex returns to its original conformation. Okay, and this complex will again inhibit the binding of um, actin from myosin. So the power stroke cycle or the cross bridge cycling ceases, and the muscle fiber returns to its normal relaxing state. Okay. Now, what is a motor unit? Now, a motor unit is a functional unit consisting of a motor neuron and the skeletal muscle fiber that innervates. From here, hopefully, you are beginning to see the intricate relationship between the nervous system and the muscular system. Okay, so the motor neuron is a type of nerve cell that transmits signals from the central nervous system to the skeletal muscle fibers, and it is the motor neuron that serves to transmit the signals from the brain and the spinal cord to give instructions to the skeletal muscle fibers that they need to contract. Okay? Each motor neuron can innervate muscle fibers, can innervate multiple muscle fibers. Okay? So you can have one motor neuron innervating just one muscle fiber, or you can have a motor neuron innervating multiple muscle fibers. And the number of muscle fibers innervated by a single motor neuron varies depending on the function of the muscle. So, for example, in muscles that require very fine movement or very fine motor control, for example, in the fingers, a single motor neuron innervates only a few muscle fibers. Whereas in areas of the body that do not actually require a lot of precise control, like in larger muscles, like in the legs, okay, a single motor neuron may innervate um, thousands of muscle fibers. Okay, so skeletal muscles, as mentioned, are innervated by motor neurons, and again, they transmit signals to skeletal muscle fibers, which cause them to contract. Okay? The process of skeletal muscle innervation involves several steps. This is more or less similar to um, muscle contraction earlier. So you have motor neuron cell bodies, which are the cell bodies of motor neurons located in the anterior spinal cord, and they receive signals from higher up in the central nervous system, particularly the brain. Okay, so they have axons which extend from the spinal cord to form synapses with the muscle fibers. Okay, and then you have the neuromuscular junction, which is the space between um, the axon of the motor neuron and um, the muscle fiber. Okay, so we know that once depolarization spreads to the axon, um, you have acetylcholine that is released into the neuromuscular junction, which facilitates the spread of action potential in the sarcolemma and in the T tubules reaching the sarcoplasmic reticulum, where calcium is released into the sarcoplasm. Okay, and um, this will result in a series of steps that lead to contraction and relaxation of the muscle. Now, um, rigor mortis is something that we see in dead people. We see this all the time in autopsies, okay, or even in um, normal dead people. So this is a change that takes place when a person dies, usually taking place several hours after death. It is a condition wherein the muscles become stiff, they become hard and rigid, making it very difficult to move the limbs or the joints. Okay? Now, rigor mortis occurs as a result of a lack of oxygen. Because there's less oxygen, ATP is not produced, and it causes the release of calcium ions in the muscle fibers. Remember, when cells die, um, they release calcium. Okay, the calcium that is released triggers cross-bridge formation between actin and myosin fibers. However, since ATP is no longer produced, the relaxation part of the power stroke does not take place, which means that the muscles become hard and rigid and they do not relax. Okay. Um, Rigor mortis usually tends to develop several hours after death, and it is maximal at around 12 to 24 hours, and then it usually disappears after 1 to 2 days when the muscles start to break down. Okay, rigor mortis is a valuable tool for us as pathologists in determining the time of death in cases wherein we don't really know when the patient died. Okay, so the degree of rigor mortis can provide clues about when the patient died, and this helps down narrow the time of death. Now, so sensory receptors in the skeletal muscles, known as muscle spindles, okay, play a critical role in monitoring or sensing muscle contraction. So muscle spindles are specialized sensory organs that are embedded within muscle fibers, and they help detect changes in muscle length and tension. Muscle spindles consist of several muscle fibers or specialized muscle fibers which are surrounded by a capsule and connected to sensory neurons. Okay, so they are muscle fibers that are connected to neurons. When the muscle fibers contract, they stretch the muscle fibers and the capsule of the muscle spindle. And this stretch activates the sensory neurons, which will then send signals to the CNS. Okay? So the CNS uses the information that it gets from the muscle spindles to monitor muscle length and tension and to regulate muscle contraction. They are integrated with signals from other sensory receptors such as the Golgi tendon organs, which monitor changes in muscle tension caused by contraction, and in joint receptors, which monitor changes in joint position and movement. Okay, so together these sensory receptors provide important feedback to the central nervous system about the position and movement of the body, allowing for precise control of muscle contraction and movement. The CNS can then make adjustments to the activation of motor neurons and muscle fibers to maintain the desired level of muscle tension and movement. And this plays a role in involuntary muscular control, for example, posture, etc., etc. Now, myasthenia gravis and botulism are both neuromuscular disorders that affect the transmission of signals between the motor neurons and skeletal muscle fibers. So, myasthenia gravis is an autoimmune disorder that is characterized by muscle weakness and fatigue, particularly in the muscles, in the smaller muscles. Okay, they control eye movement, facial expression, and swallowing. What happens here is that the immune system produces antibodies against the receptors to acetylcholine, okay, which reduces the number of receptors available for binding by acetylcholine to facilitate contraction. So as a result, the transmission of signals from the motor neurons to the muscle fibers is impaired, which leads to muscle weakness and fatigue. Now, this becomes particularly dangerous when um, the levels of antibodies against the acetylcholine receptors become so high that they will also involve the diaphragm. So, in very severe cases of myasthenia gravis, you will have patients who can no longer breathe properly because their diaphragms are paralyzed. Okay? And we know that the diaphragm is a major inspiratory muscle. Okay? Now, botulism, on the other hand, is a type of food poisoning that is caused by the ingestion of a neurotoxin produced by a certain type of bacteria called Clostridium botulinum, which is a relative of Clostridium tetani, or the organism that causes tetanus. Okay? The botulinum toxin binds to the presynaptic membrane of the motor neurons, preventing the release of acetylcholine into the synaptic cleft. So, in myasthenia, you will have antibodies that prevent the binding of acetylcholine in the postsynaptic receptors, or receptors in the muscle portion. In botulism, you will have a toxin that inactivates the presynaptic membrane, or the neuron end, which inhibits the release of acetylcholine. Okay? This leads to a condition known as floppy paralysis, where the muscles do not contract, they become floppy and they become weak. Okay? So botulism is usually life-threatening. We usually get botulism in improperly canned food, okay, or in honey. Okay, so this is the reason why you should refrain from giving babies honey because you may transmit botulism. However, botulism is also used for therapeutic purposes. Okay, we know that botulism is used in Botox, hence the name botulinum toxin, and they prevent wrinkles by paralyzing that small muscles in your face. So you get very small amounts of botulinum toxin. Okay. So while myasthenia gravis and botulism both affect the transmission of signals between motor neurons and skeletal muscle fibers, they have different, although related, underlying causes, and they require different treatments. Okay. So this slide shows us the different properties of skeletal muscle as a summary. Cardiac muscle is a specialized type of muscle tissue that is found exclusively in the heart. It has several unique histologic properties that distinguish it from other types of muscle tissue. Um, Like skeletal muscle, it was also striated, and it is due to the organization of actin and myosin filaments into sarcomeres. This gives cardiac muscle a banded or striped appearance under the microscope. Okay, this is a scanner or a low power view. This is a much closer view. So here you can see that they have a branched structure. Okay, they are connected by intercalated discs seen here. Okay, these are the intercalated discs, and these actually represent gap junctions and desmosomes that allow for rapid transmission of electrical signals from one cell to the adjacent cell. Okay, one cell to the adjacent cell. Okay. This branching structure allows cardiac muscle to work as a coordinated unit and helps to ensure efficient and synchronized contraction of the heart. Okay. Unlike skeletal muscle, cardiac muscles contain a single nucleus. Okay, they are uninucleate, meaning they only contain one nucleus per cell, unlike skeletal muscle, which have multiple. Also, unlike skeletal muscle, the nucleus is now located centrally. Okay, in contrast to skeletal muscle, where the nuclei are located in the periphery. They tend to have a high density of mitochondria, which makes sense because you need the energy for the constant contraction of the heart. Okay, it would be dangerous if the heart will not produce enough energy to power its own actions. Okay, the mitochondria is located around the nucleus and below the cell membrane. Okay, now the heart also has an endocardial layer or an endocardium. Okay, which is a thin layer of connective tissue that separates the muscle fiber from the blood in the heart chamber. So the endocardium actually um, serves as protection to the cardiac muscle fibers para dili siya mugasgas sa turbulence of the blood. Okay? So these properties allow cardiac muscle to function as a highly specialized and efficient tissue for pumping blood throughout the body. Again, here's a summary of the characteristics of cardiac muscle. Now, smooth muscle is the third type of muscle tissue that is found in the walls of hollow organs and structures such as blood vessels, the gastrointestinal tract, and the respiratory tract. Um, it has a non-striated appearance. It does not have a banded appearance, unlike skeletal and cardiac muscle, because it does not have the orderly arrangement of actin and myosin filaments. Okay? The cells, aside from being non-striated, also are spindle in shape. Okay? They are elongated, and they have a single nucleus which is located in the middle. Okay? There is also a lack of sarcomeres, um, in contrast to skeletal and cardiac muscle. Okay? Um, then, smooth muscle cells have what are known as dense bodies, which are structures that anchor the actin filaments in place and allow for the transmission of force between adjacent smooth muscle cells. Um, these dense bodies give smooth muscle its characteristic hill and valley appearance. Hill and valley appearance. Okay. Smooth muscle is innervated by the autonomic nervous system, which allows for the regulation of smooth muscle contraction and relaxation. Smooth muscle can be classified into two types based on the organization of muscle cells into functional units. So, multi-unit smooth muscle are innervated by separate nerve fibers and act as independent units. So they act independently of one another. This type of smooth muscle is found in structures such as the iris of the eye, the arrector pili muscles, which are the muscles that control your hair follicles, and it is also found in smooth blood vessels. Multi-unit smooth muscle is controlled by the autonomic nervous system and can exhibit fine control over contraction and relaxation. In contrast, unitary or single unit smooth muscle are organized into functional unit called syncytia, okay, in which many cells are connected by gap junctions and they act as a coordinated unit. They cannot contract independently. They contract together. So single unit smooth muscle is found in the walls of hollow organs such as the uterus, the bladder, the digestive tract. And this is also controlled by the autonomic nervous system but can exhibit synchronized rhythmic contractions, unlike multi-unit smooth muscle which act independently of one another. The distinction between multi and single unit smooth muscle is based on the degree of innervation and the organization of smooth muscle cells within the tissue. Okay? Here are photos. So this is the smooth muscle of the stomach. Okay? You have the lining epithelium here, and you have the muscle layer here. Okay? Here you have muscle, muscle, muscle. Okay? Finally, a summary of the different properties of smooth muscle. The muscular system is closely intertwined with the nervous system because the nervous system controls muscle movement. When we want to move a muscle, the nervous system sends electrical signals through neurons to the muscle fibers, causing them to contract and create movement. So this process is known as neuromuscular control. Okay? The nervous system also receives feedback from the muscles about their position, tension, and movement, which is important for coordinating and adjusting movement, for adjusting posture. This feedback is sent through sensory neurons that connect the muscles to the nervous system, allowing for real-time adjustment of posture and fine-tuning of movements. Okay? And additionally, the muscular system plays an important role in maintaining posture in balance, which requires continuous monitoring and adjustment by the nervous system, um, and regulation of muscle tone, which is important for maintaining muscle readiness and preventing injuries. Okay? So the nervous system can be divided into two main divisions, the CNS and the PNS, the central nervous system and the peripheral nervous system. The central nervous system includes the brain and the spinal cord, and it is responsible for receiving, processing, and interpreting sensory information from the PNS or the peripheral nervous system, and for sending out motor commands to the peripheral nervous system. Now, the PNS or the peripheral nervous system includes all the nerves outside the brain and the spinal cord. Okay? It can be further subdivided into two main divisions, the somatic nervous system and the autonomic nervous system. The somatic nervous system consists of nerves that control voluntary movements and transmit sensory information from the brain to the CNS. Okay? The autonomic nervous system, on the other hand, regulates involuntary activities. So somatic is voluntary, autonomic is involuntary. Okay? It is the autonomic nervous system is further subdivided into the sympathetic and parasympathetic nervous system. So the sympathetic nervous system is responsible for the fight or flight response, which prepares the body for action in response to stress or danger. While the parasympathetic nervous system is responsible for the rest and digest response, which helps the body conserve and restore energy and maintain normal bodily functions. Okay? So overall, the divisions of the nervous system work together to regulate and coordinate the body's activities in response to internal and external stimuli. So as with muscular tissue, the nervous system is surrounded and protected by several layers of connective tissue, which provide support and insulation to the nerves. So the main connective tissue investments of the nervous system are the epineurium, the perineurium, and the endoneurium. Okay? The epineurium is the outermost layer of the connective tissue that surrounds an entire nerve. Again, it's composed of dense, irregular connective tissue. So this is analogous to the epimysium of the muscular system. It provides protection and support to the nerve and helps to maintain the shape of the nerve. Okay? Deep to the epineurium is the perineurium, which is a layer of connective tissue that surrounds each nerve fascicle. Okay? It is analogous to the perimysium. It is made up of specialized cells called perineural cells, which form a barrier that separates the nerve fibers from the surrounding tissues and fluids. Okay? And then finally, you have the endoneurium, which is the innermost layer of connective tissue that surrounds each individual nerve fiber, and this is analogous to the endomysium in muscles. It is composed of delicate reticular fibers and provides insulation and support to the nerve fibers. So these connective tissue investments work together to protect and support the nerves and to maintain the integrity of the nervous system as a whole. They also play a role in the exchange of nutrients and gases, particularly oxygen, as well as the waste products of nervous tissue for removal into for removal out of the body. Okay. So you have the endoneurium, the perineurium, and the epineurium. Okay. The CNS contains several types of specialized cells that work together to regulate and coordinate the body's activities and responses to stimuli. So the main cell types of the CNS are as follows. Number one, you have the most important cells called the neurons. Okay, they are specialized cells that serve to transmit electrical and chemical signals throughout the nervous system. Okay, they are the basic units for communication and processing in the CNS and are responsible for sensing, interpreting, and responding to various stimuli. Now, you also have a different family of cells known as glial cells. They are also known as neuroglia or simply glia. These are non-neuronal cells that provide support and protection to neurons in the CNS. So these include astrocytes, which are the most abundant type of glial cells. You also have the myelin-producing cells, oligodendrocytes, and Schwann cells. Okay. You also have the resident immune cells of the CNS, known as the microglia. And you have the ependymal cells, which are involved in the production and regulation of the production of CSF or cerebrospinal fluid, which serves to cushion and protect the brain and the spinal cord. So the neurons work hand in hand with the glial cells to form the complex network of cells that make up the CNS. Okay? So this is how neurons look like. Okay? We'll talk about the different parts of the neurons in a bit. This is a picture of the spinal cord, cross section of the spinal cord. The different parts of a neuron work together to receive, process, and transmit information throughout the nervous system. And this allows for the complex and coordinated function of the body. So, the neurons or nerve cells are the fundamental building blocks of the nervous system. And again, they are highly specialized in that they are designed to receive, process, and transmit electrical impulses. The main parts of a typical neuron include the cell body, and this is also known as the soma or the pericardion, excuse me. The soma or the cell body is the main part of the neuron that contains the nucleus and other organelles that are responsible for carrying out the basic functions of the cell. Dendrites, on the other hand, are branch structures that extend from the cell body and receive incoming signals from other neurons or sensory receptors. They play a key role in integrating and processing these signals before transmitting them to the cell body. Okay, so they receive stimuli, they receive impulses. The axon, on the other hand, is a long, thin fiber that extends from the cell body and transmits outgoing signals to other neurons or effector cells such as muscles or glands. So it is uh, responsible for conducting impulses away from the body. Okay, the axon terminates into what is known as the axon terminal. Okay, and this is the part of the axon, the terminal part, that forms a synapse or a junction with either another neuron or an effector cell, like a muscle. Um, it is in the axonal terminal where um, you will have specialized vesicles for neurotransmitters, which are chemical messengers that are transmitted across the space between one neuron to another. Okay? Now, another structure is the myelin sheath, which is an insulating layer that surrounds axons and helps to speed up the transmission of electrical signals. Okay, it is formed by specialized cells called oligodendrocytes in the CNS and Schwann cells in the peripheral nervous system. Okay, so what forms the myelin sheath in the brain and the spinal cord? Oligodendrocytes. What forms the myelin sheath in the peripheral nervous system? Schwann cells. Okay. Astrocytes are a type of glial cell that is found in the CNS. So they are known as astrocytes because they have the appearance of a star, okay, due to the presence of dendrites emanating from the cell body. They are the most abundant type of glial cell, as mentioned, in the CNS, and they play a critical role in maintaining the health and function of the nervous system. Astrocytes have many functions, and these include providing physical and metabolic support to neurons. They help to maintain structural integrity of the nervous system. They provide metabolic support to neurons by regulating the exchange of nutrients and waste products between the neurons and the blood vessels. They also form a structure known as the blood-brain barrier, which is a specialized barrier that separates the blood circulation from brain tissue. This is crucial because it facilitates protection from of the brain from toxins, from pathogens, and other harmful substances. Um, they also play a part in regulating neurotransmitter levels. Okay, they can take up excess neurotransmitters, for example, glutamate, and they convert them into less harmful products. Astrocytes can also respond to injury and inflammation in the nervous system by proliferating and releasing certain molecules that can limit the extent of damage and promote repair of tissues. Oligodendrocytes are another type of glial cells in the central nervous system that produce myelin, which is a substance that wraps axons and help to insulate and speed up the transmission of electrical signals between neurons. So in the CNS, each oligodendrocyte can myelinate multiple axons, forming multiple segments of myelin sheaths. So oligodendrocytes are important for efficient neuronal communication in the brain and the spinal cord. Um, Damage to oligodendrocytes or the myelin sheath can lead to certain conditions such as multiple sclerosis, wherein the immune system attacks and damages myelin in the CNS. Okay, so this is the axon, the dark line here in the middle, and the gray, lighter gray structure surrounding it is actually the myelin that's produced by oligodendrocytes. Schwann cells are another type of glial cell that is found only in the peripheral nervous system. So, like oligodendrocytes in the CNS, Schwann cells also produce myelin. However, each Schwann cell can only myelinate a single axon, unlike oligodendrocytes. This means that multiple Schwann cells are required to myelinate a single nerve fiber. Okay? So, in addition to myelination, they also support axonal regeneration after injury, and they help to maintain the health and function of the nerves or the neurons they surround. Um, Damage to Schwann cells and myelin sheath can also lead to other neurological disease, such as an example is a condition known as Charcot-Marie-Tooth disease, which is a hereditary disorder that affects the peripheral nerves, and this causes muscle weakness and muscle degeneration and atrophy. Okay? Microglia are specialized cells of the immune system that are found in the CNS, including the brain and the spinal cord. They are the smallest of the glial cells, and they account for about 10 to 15% of the cells in the CNS. They have important functions, particularly immune surveillance. They also play a role in phagocytosis, which is when cells engulf or eat up cellular debris and other foreign substances like bacteria. Um, Microglia also promote tissue repair and regeneration. Um, Microglia have been implicated in several diseases as well, including Alzheimer's disease, Parkinson's disease, schizophrenia. Okay, they also play a role in normal brain development and plasticity, as well as synaptic pruning, or the process whereby unnecessary synapses are removed or eliminated during development. Ependymal cells, on the other hand, are found in the CNS, and they line the walls of fluid-filled cavities known as ventricles. Okay? They have a lot of important functions, but the most important is the production and regulation of cerebrospinal fluid, which we know is important in protecting the brain and the spinal cord. CSF is also important in the circulation and removal of waste substances. Okay. Now, a synapse is a specialized junction between two neurons or between a neuron and a target cell, for example, a gland or a muscle cell. And they are the fundamental units of communication in the nervous system and allow neurons to transmit information to one another and to other cells in the body. Synapses can be classified based on their location, function, and structure. So for example, synapses can be classified as axodendritic, or between an axon of one neuron and the dendrite of another, or they can be classified also as axosomatic between the axon of one neuron and the soma or the cell body of another, or axonic, between the axon of one neuron and the axon of another. They can also be classified based on the type of neurotransmitter involved, such as cholinergic, um, acetylcholine release, dopaminergic, dopamine release, or serotonergic, or serotonin release. So there are two main types of synapses, chemical synapses and electrical synapses. Chemical synapses are the most common type, and they involve the release of chemical neurotransmitters from a presynaptic neuron. So when an action potential reaches the end of the axon, it triggers the release of these neurotransmitters. The neurotransmitters that are released into the synaptic space leads to changes that facilitate binding of the neurotransmitter to the postsynaptic cell. Okay? Electrical synapses, on the other hand, is where electrical current flows directly between the cells via specialized communication channels known as gap junctions. So, electrical synapses facilitate rapid, much more rapid bidirectional communication between neurons. Electrical synapses are far less common than chemical synapses, but are found in certain types of the brain and in other organs, such as the heart and the smooth muscle. So these are the common types of neurotransmitters that I'm sure most of us have been exposed to during our physiology classes. So nerve fibers are long, thin, thread-like extensions of nerve cells that transmit electrical signals. And the term nerve fiber collectively refers to two main types or two main parts of the nerve cell. They may refer to the axons or they may refer to the dendrites. Okay? So the myelin sheath is a protective and insulating layer that covers the nerve fibers. So it's the wrapper around the nerve fibers. And again, it is composed of a fatty substance known as myelin, again produced by oligodendrocytes in the CNS and Schwann cells in the peripheral nervous system. This myelin sheath serves important functions. First, it helps to insulate the nerve fibers, and by insulation, we mean that they do not conduct the the electrical impulse. But because of that, it actually serves to increase the speed and efficiency of electrical signal transmission. Okay? And this is because, as mentioned, the myelin sheath acts as an insulator. It prevents electrical flow. Okay? So you have to understand that the myelin sheath is not continuous. Okay, as what we will find out later, there are actually gaps in the myelin sheath known as the nodes of Ranvier, and this facilitates the rapid jumping of the electrical impulses from one end of the nerve fiber to the other. But before that, we have to also recognize that the myelin sheath helps protect nerve fibers from damage and degradation. So it plays a part in structural support. And this prevents axons from becoming compressed or damaged due to movement or other physical stress. Okay? Now, as mentioned, the nodes of Ranvier are small gaps or breaks in the myelin sheath that surround the axons of neurons in the nervous system. So they are found in regular intervals along the length of the axon and are important for the efficient transmission of electrical signals across the axon. The myelin sheath helps to insulate the axon and speeds up the transmission of electrical signals by preventing the signal from leaking out of the axon. However, because the myelin sheath is continuous, it makes it difficult for the electrical signal to jump from one section to another. And This is facilitated by the presence of gaps, okay, along the nerve fiber called the nodes of Ranvier, which allows the nerve fibers to um, facilitate the conduction. So here we have again a segment of a nerve fiber. Here you have the axon or the dendrite. You have the nerve fiber here, the dark structure in the middle, and you have the myelin sheath, which is the dark gray substance surrounding um, the nerve fiber. Now, in regular intervals, we see um, these gaps, like this one here, this one here, this one here, and this is known as the nodes of Ranvier. Note that the myelin sheath is an insulator, so it actually prevents um, the movement of electrical impulses because it serves as an insulator. However, because you have these gaps here, the nerve fi- the nerve the impulses actually jump from one gap to another, making it quicker for the transmission. Okay, so for example, here you have one node of Ranvier here, one node of Ranvier here, another here. So instead of going through the whole thing, the nerve impulses actually jump from one node to another, making the transmission uh, much more quick, much quicker than if the nerve impulse has to travel through the entire length. So this jumping of nerve impulses is known as the saltatory conduction. Okay? So the CNS is composed of white matter and gray matter, and they have distinct characteristics. So the gray matter is composed of cell bodies, dendrites, and axonal terminals of neurons, as well as glial cells to support and protect the neurons. So gray matter is given the name because it has a gray-pink color, which comes from the dense clustering of cell bodies and other structures. It is usually located on the outermost portion of the brain called the cortex, and in various clusters of nuclei throughout the brain and the spinal cord. Okay? So gray matter is responsible for thinking. It is responsible for processing and integrating information, as well as for initiating and controlling motor movements. White matter, on the other hand, is composed mostly of myelinated axons, which are long, thin projections, okay, which was explained earlier. White matter appears white due to the presence of myelin sheath. So, whereas gray matter is for processing information, white matter serves as a communication network, allowing different regions of the brain and spinal cord to communicate more efficiently. Because we know that white matter is composed mostly of myelin. In general, the more complex the brain function, the more gray matter is required. However, white matter is required in areas of the brain which is necessary for efficient and rapid communication. Okay? So this is a picture of the spinal cord and the peripheral nervous system. So the brain gray matter is located at the cortex or the periphery of the cerebrum and the cerebellum. The white matter is located underneath. Okay? The CSF is a clear, colorless fluid that fills the ventricles of the brain, the central canal of the spinal cord, and the subarachnoid space, which surrounds the brain and the spinal cord. It is produced in the ventricles of the brain by specialized cells in the choroid plexuses called um, ependymal cells, and the CSF provides a cushioning and protective effect. Okay, so CSF has a protective function. It acts as a shock absorber, which cushions the brain and the spinal cord. It also provides buoyancy, which helps reduce the weight of the brain, making it easier to support and to move around. And then CSF is also important in the delivery of nutrients and oxygen, as well as the transport of hormones throughout the body. The CSF also plays a role in the immune response, helping to protect the brain and the spinal cord from infection and inflammation. And CSF helps to regulate the pressure inside the brain. So you can have disorders in the production and the circulation or absorption of the CSF, and these can have significant neurologic consequences. So, for example, we have a condition known as hydrocephalus, wherein there is an abnormal accumulation of CSF in the brain, which leads to increased pressure and damage to the brain tissue. Okay? You can also have inflammation of the wrapper of the brain, known as meninges. And this can, the inflammation or the infection can spread to the CSF, and this can lead to potential serious complications in the CNS. Nerve endings are specialized structures located at the distal end of nerve fibers, where they communicate with other cells or with the environment, like the skin, the fascia, the muscles, joints, and the tendons. And they contain receptors that are specialized proteins that detect specific type of stimuli, such as pressure, temperature, pain, or chemicals. There are several different types of nerve endings and receptors. These include mechanoreceptors that respond to mechanical stimuli. So they respond to pressure, they respond to stretch. And we see mechanoreceptors in the skin, joints, muscles, and other tissues. And they are responsible for a sense of touch, pressure, vibration, and proprioception, or the ability of our body to sense where our body is in space or our positioning. Another type of nerve ending receptor is a thermoreceptor, and they respond to changes in temperature. They allow us to sense heat or cold. Okay? Nociceptors or nocireceptors are pain receptors. They are activated by molecules that are released during tissue damage and inflammation. You also have chemoreceptors, which respond to chemical stimuli such as taste and smell. Okay? And photoreceptors, which respond to light. So examples of mechanoreceptors include Meister's corpuscles and Pacinian corpuscles, which are responsible for touch and pressure detection. Okay? So these are free nerve endings in the skin. These facilitate detection of pain. Okay? This one is a Pacinian corpuscle, okay? Onion-like. And they facilitate detection of deep pressure. Okay, or deep touch. Again, a Pacinian corpuscle. So these are examples of mechanoreceptors or sensory receptors in the skin and um, the deeper tissues. Okay, so hopefully you were able to get something out of this discussion. If you have any questions, since this is a recorded lecture, kindly do not hesitate to post your questions in our Telegram group so I can answer them and everyone in the class can see. Thank you very much for your attention. I will see you next Friday. Good luck for your P1 exam on Thursday. Bye bye.